Welcome, everybody. Welcome to the FRAXA webinar series. I'm Dr. Mike Tranfaglia. I'm the Medical Director and Chief Scientific Officer of FRAXA Research Foundation. I just wanted to welcome you to today's presentation, where we will be featuring the folks from Healx. Healx is a repurposing company in Cambridge, England. And FRAXA has been working very closely with Healx for several years now to repurpose available drugs and combinations thereof for Fragile X. But they also do a lot more than just Fragile X. And so to kick things off, I'd like to introduce you to Dr. Dave Brown, who is the chairman and co-founder of Healx. He'll be talking to you about Healx's methods used to select drugs for preclinical testing in models of Fragile X. Dave?
Okay, thanks, Mike. I'm going to share my screen and slides. Right, so welcome everybody. As Mike said, I'm co-founder and chair. I was chief scientist actually as well, multitasking at the time when Mike first approached us in 2017, and worked on the Fragile X project for a couple of years and still doing a little bit of work on it. It was one of our biggest efforts, and I think looks quite successful. After me, you're going to hear about the animal testing and then the clinical trials that are being lined up, but I'm going to talk about how we initially approached this and came up with predictions of drugs that could work. So what I've got here is a diagram that's used in the pharmaceutical industry, just showing the length of new drug discovery and the complexity. I don't know if you can see my pointer, but on the left-hand side where it says idea, people usually propose to work on a disease and make a guess at a target biochemical to go for. And I think that's the way most Fragile X research is being done, including many drugs that have failed in the clinic, unfortunately. Having selected a target, then people screen chemicals against it to find a lead molecule, then years and years of work optimizing that lead molecule to get the properties that are required. And this whole process takes sort of 2 to 5, 6 years normally. Then there are regulatory studies that need to be done to show the drug is safe and can be dosed to humans, and scaling up the chemical synthesis. And then clinical trials that are split into phase 1, phase 2, phase 3. Phase 1 is essentially safety and how the body handles the drug. It's not necessarily efficacy being measured there. But phase 2 is the beginning of dosing to patients, and phase 3 is scaling up. And then registration of the drug and take it to market. That whole process I've spent decades doing, and actually got several drugs to market myself. But you know, this takes 10 to 15 years for most projects. It's a very, very long process, very expensive, and an extremely high failure rate. At Healx, we're trying to change that, trying to make it faster and cheaper. And the methods I'm going to show you basically replace those 4 steps with a single parallel process. We then replace these 2 steps totally. And by doing that, by repurposing old molecules rather than discovering new molecules, the whole process can be done in about 3 years rather than 12 years. This is summarized in the literature. It's not Healx data. It's summarized from many other companies and studies. Cost to develop of a new drug is enormous. It's in the billions. This is 1,800 million, 1.8 billion, including all the failures. The evidence is that for repurposing, it can be done for a fraction of that, much smaller. And the probability of success there is put at 5%. Personally, I think that's optimistic. I think the overall success is lower than 5% actually for new molecules, whereas for repurposing, it should be about 1 in 4, 1 in 5 at worst will make it through. So quicker, cheaper, safer altogether. So the other factor here is that many diseases are very complicated. You're very familiar with Fragile X and the broad range of phenotypes, and the chance that a single drug is going to treat the several symptoms that most patients have is not very good. And we know that for most diseases, patients are given 2 or 3 or 4 or even 5 or 6 drugs. It can be very difficult to develop 2 new drugs for one disease. I actually tried this in the days when I was working for Pfizer way back in the past, and that was a lesson. You know, thankfully it failed after a couple of years. It was getting very difficult and extremely expensive. But if one of those drugs in a combination is a repurposed safe drugs, it makes the whole thing a lot easier. And that's the way we're approaching it in Healx. So this one's a little bit complicated. Just saying, we're an AI company. We have these things called algorithms. It's just computer programs, basically. We have several for looking for single drugs that might work, and we have several for combinations of 2 or 3 drugs that might work. If you go through all the existing known drugs that are registered in the USA and elsewhere, there are a large number of drugs around the world. And if you you can match those to the disease in some way and come up with a monotherapy prediction, that's not too difficult. But by the time you get to combinations of 2 or 3 drugs, it turns into an enormous number of possibilities, and this is much more difficult. And that's what we're trying to do, and we have had some success with that. So we're trying to predict monotherapies to start with, and then combinations of drugs that might work against the disease. I put a very simple diagram here, which illustrates one of our methods. And this was the one that we used with Mike way back in 2017, I think it was, when we first started. The company was quite young then, and we didn't have so many methods, but this was our sort of base method at the time. If you imagine that these are genes being expressed in various levels or underexpressed at various levels, the normal non-disease state would be the flat line here. Fragile X would be some genes being overexpressed, some genes being underexpressed. What we're looking for is a drug that really collapses those overexpressed genes and increases the underexpressed genes and returns it to something like the normal state. Very simple concept, but it actually works quite powerfully. We call this sort of disease drug signature matching. DGEM is our terminology for it. So this was the method, well, we actually used 2 methods at that time, but this was the method that gave us about 80 or 90% of the hits of the predictions at the time. So the important thing here is that what we're doing is hypothesis free. If you go back to that diagram I had here of the drug development process, where people are guessing at a target that might work against a disease when you get way down here. We're not doing that. We're taking the disease state here, matching it to a drug, almost working backwards, matching it to a drug, and then working back to the targets in which it may be working. And that's proved very effective. So this is a hypothesis-free approach based on human patient tissue, or it could be these cell constructs, and what we call transcriptomics. This is what the expression of the DNA is called transcription of the DNA. That's where this jargon comes from. And we're reversing the disease signature, which is what I mean by this collapsing back to normal, and selecting a drug. And once we've done that, found a match, we then look at the drug, look at what's known about the disease, and see if we can figure the mechanism of action of the active drug. That's where a lot of pharmacology expertise comes in. It gets very interesting at that point. So we started this towards the end, 4th quarter of 2016. Over the winter 16-17, we did our in silico predictions, came up with a number of predictions, and sent the report off to Mike. I remember he immediately responded, literally a few hours on the next day, saying, "Oh my God, you predicted in here some drugs we've already tested and we've proved that they're active." Metformin was one of them. And we didn't know, it hadn't been published. So he was quite impressed that we had predicted that with no knowledge at all. And so we then had confidence to test the other predictions. FRAXA have the drug validation initiative with Patricia Cogram in Chile. We sent them off there, and we had a number of them showing quite good activity. We actually published this wrote it up over the winter of 2017-18, and it was published online. And you can access this publication to see exactly how we went about that with Mike as the lead author. We then started a combination drug project. And this is where it gets very exciting. And my God, it gave me a headache trying to understand all the combinations that were being predicted. But we came up with some interesting ones, both two drugs combined and drugs and nutraceuticals combined, that began to show quite interesting activity in the Fragile X mouse model. What we're looking for here is drugs that are partially active alone becoming fully active when you get them together. So we're seeing either an additive or a synergistic effect. And after I finish, Ivan is going to show the data for some of the drugs that we predicted. We're now moving one of those combinations through to clinical trials in the US, and later Tony Hall will be describing the clinical planning for that combination as well. This is my last slide. It's awfully complicated, but this is a neuron and in the nervous system. And what I've got here in red in these boxes are the mechanisms that have been tried in the past for Fragile X, the GABA receptor, mGluR5, ampakine receptors, etc. Each of those have been selected by the old way of doing drug discovery, as I call it. In other words, make a guess at what the deficit is and try and fix it. As I said, we didn't do that. What we did was find something that reversed the disease signature and then work back to mechanisms. And we probably have multiple mechanisms actually in the drugs that we're picking out. So what we call Healx 01, a drug is sort of a core drug in the combinations we've been testing. It modulates several targets and pathways. And I've identified those in green here. So we're getting multiple hits just with that one drug down here, very different from the ones that have failed in the clinic. And we've now added a second drug to the combination that hits other targets as well. So there's a good chance, I think, that we will get better effects in the clinic than we've seen with the single mechanism drugs in the past. That is where I finish, and I'll hand over to Ivan, who will show you the data for the compounds we predicted.
And I just wanted to mention to everybody that we'll be taking questions at the end after all three presentations, but you can start by registering your questions in the chat function of Zoom, and we'll take them in order after all the presentations are complete. So on to you. Ivan Angulo Herrera is the Associate Director of Pharmacology at Healx, and he's going to be talking about the preclinical results from the FMR1 knockout mouse model. Ivan?
Can you see my screen now?
Yes.
Well, thank you, Mike, and thank you, Fragile X Research Foundation, for the opportunity to present in this seminar and present our results. So today, just as we just discussed, I'm going to be presenting the preclinical result that we have tested in the FMR1 knockout model for Fragile X. So to give a little bit of context on the disease and the preclinical model, so as you all know, so the mutations in the Fragile X mental retardation protein cause a wide spectrum of symptoms in humans, from seizures, sleep disorders, anxiety, irritability is the #1 cause of autism, and also cognitive impairments and intellectual disability. So it's known that this protein, FMRP, is related with translation of mRNA and controlling synaptic function by the way it controls translation and participates in translation of other proteins that are involved in synapses. So when this protein is missing in the brain, there is a normal expression of protein that normally are very, very tightly regulated. The way people have been modeling the disease, so in mice on fruit flies, is just making deletions of this gene in the mice or the equivalent in the flies, and observing what are the changes that behavioral and more molecular changes that these models has in their brains. So in both the Fragile X patients and the FMR1 knockout mice, there are alterations of the density, size, and shape of the dendritic spines. That is one of the common feature of these mice. And also there are several aspects of the neuropsychiatric symptoms in the Fragile patient that can be recapitulated in the mouse model. And when we look at different behavioral tests, like to look at impairments of memory and learning, hyperactivity, anxiety tests, and social interaction of the mice, all these tests to some extent can recapitulate what is happening in the actual individual with the syndrome. So talking about the mouse model that we've been using, so this is the FMR1 cow2 mouse that was developed by the Dutch consortium in 2016. This is a mouse that lacked the expression of the protein and also the messenger RNA of the FMR1. And this mouse has been also characterized by a battery of behavioral tests at the GeneDDI lab in Chile by Robert Deacon and Patricia Cogram. So as you can see here, when they characterize this mouse, you can see that they have different impairments in different behavior. For example, when we're talking about memory, for instance, if we compare how the wild type animals are able to recognize a novel object, if we look at the knockout animals, is basically look at a novel and a familiar object the same amount of time. So it is a deficient memory to look at novel object versus familiar object. Similar when you compare wild type animals with the cow animals for tests like fear conditioning, you see that they are less able to remember a moment of fear. These animals also have some impairment in sociability when they are exposed to a novel mouse. In comparison to the wild type animal that spend a significant amount of time with the novel mouse, these animals basically spend the same amount of time with the familiar versus the novel mouse. Also, these mice are more hyperactive. They explore more and travel more within the cage than the wild type animal, as you can see in this different in the open field test. And also these mice have anxiety. When they are exposed to a novel food, they get anxious because they have this fear if the food is going to be good for them or not. So the time that they spend to take the new food is longer than the wild type animals. Also, they have repetitive behavior by increased level of self-grooming. They have deficiencies in building nest, and they're also more aggressive. The time that they spend between aggression episodes with a novel mouse is shorter than the wild type mice. So there are several potential behaviors that have been already characterized in the FMR1 mouse compared to the wild type that you can use to test drug to see if they are able to revert those effects. So when we look at the repurposed drug that we wanted to test, so as Dave was describing before, we started the collaboration with FRAXA Research Foundation, and we identified several drug candidates as potential treatment for Fragile X. One of the candidates that we identified was fenformin. That is a biguanide antidiabetic agent that was used a long time ago to lower blood glucose levels in human, but the drug was withdrawn due to some safety concerns. But there's a very similar drug with the same mechanism of action, which is metformin, that is widely prescribed as a medication for type 2 diabetes, can be administered orally, and have very good PK profile, and is more safe drug. So when we found this fenformin, we decided to start doing testing with metformin. We didn't know about the use of metformin in the Fragile X mouse model. The only evidence that we found at the time was some testing that people were doing in the Drosophila Fragile X model in 2017. So we were quite encouraging with this finding, and we started doing some testing to sort of validate the technology and validate the prediction that we came up with. So in the first experiment that we did in the Fragile X mouse model, we dosed the animal using osmotic pump for 2 weeks. With osmotic pump, we have a continuous subcutaneous infusion of the drug. So we went with 50 and 100 mg per kilo daily on the mice. And we observed that at the higher concentration of the drug, we were able to rescue, for example, sociability to the wild type levels. We also observed in 2 different memory tests, fear conditioning and novel object recognition, that we were able to rescue at 100 mg per kilo to the level of the wild type animals, and significantly different to the knockout treated with the vehicle alone, or the knockout treated with a lower dose of metformin. Similar when we look at the test of the novel object recognition, again, at 100 mg per kilo, we were able to rescue to the wild type levels, and significantly better than the vehicle alone or the drug at a lower dose. Also, we in the same experiment, we test hyperactivity by the open field test, and we observe a reduction of the exploration of the mice at the highest dose of metformin. Also, when we look at anxiety, we observe that these mice treated with 100 mg per kilo of metformin spend less time until they eat the novel food, the same level to the wild type. And also these mice were able to build nests as efficiently as the wild type animal. So in the 6 different behaviors that we test this using osmotic pump, we observed that in the higher doses of metformin, we were able to rescue the 6 tests completely to the wild type levels. That was really, really encouraging. So we decided to perform a second experiment in which we move away from the osmotic pump and we focus on the intraperitoneal dosing. This time we stay on the highest dose, and actually we went with the drug twice a day instead of once a day, and we extend also the period of dosing to 4 weeks instead of 2 weeks. With this experiment, we wanted to confirm the behaviors that we observed before, but also we were looking at some toxicity effects of metformin by increasing the concentration of the drug and also increasing the length of the study. And what we observed in this experiment, in 7 different behaviors, we were able to rescue all of them. Sociability, we rescued the time that the knockout animal treated with metformin spent with novel mouse was exactly the same to the wild type, as significantly better than the knockout treated with the vehicle. Similar with novel object recognition, we rescue the time spent with a novel object. We also observed that the animals become less hyperactive by decreasing the exploration time that they have and the distance that they explore in the cage. They were having less repetitive behaviors. Also, the anxiety was reduced as well. The nesting capacity was increased, and the aggression, this time we test aggression again, and we observed that the time that they spent between episodes of aggression with a novel mouse was increased to the wild type level. And we didn't see any effect of toxicity on these mice with this higher dose of metformin and for this longer period of time of the experiment. Also, at the end of the experiment, we collect tissues and brain tissues and blood samples to analyze some biomarkers that are related to Healx. For example, we know that the loss of the FMR protein results in hyperactivation of the mTOR signaling pathway and the ERK signaling pathway. So we look at phosphorylation of ERK by flow cytometry from blood of this animal, analyzing from PBNCs in the plasma. As you can see here, the knockout animals treated with the vehicle has significantly increased level of phosphorylation of ERK. And when we treat the animals with metformin at 100 mg per kilo, we reduce the phosphorylation of ERK in the blood. Also, we look in the brain, in cortex and hippocampus. I'm only showing the data for cortex in here. And we can see that the phosphorylation level of ERK is increased in the knockout animals treated with the vehicle alone, but when we treat with metformin, we're able to reduce significantly the phosphorylation of ERK in these mice. So which is a really nice confirmation that metformin is working on target and also is reverting some of the characteristics, biochemical characteristics of the loss of the Healx mental retardation protein. We also look at phosphorylation of AMPK because this is another signaling pathway that is misregulated in Healx, is underactive in the brains of Healx mice. So as you can see here, when we look by Western blot in hippocampus, we see less phosphorylation of threonine 172 of AMPK, and we see an increase of phosphorylation when we treat the animals with metformin at 100 mg per kilo. So we were able to rescue the phosphorylation of AMPK with metformin. So also we look at dendritic spine morphology. It's known that the length of the spines has been reported in Healx patients and postmortem samples, and also in the mice that has increased length of the spine. So we look at that in the hippocampus regions, neurons from the hippocampus region, and we indeed observe this increase of length of the neuron of the dendritic spines that were also decreased when we treat the animals with metformin. So we have also this, not only the behavior, but we also have the biochemical characterization and the dendritic spine morphology to show reversion of different aspects of Healx in the mouse model. So overall, metformin is seemed to be very beneficial and have very beneficial effects in Healx. So the work that have been done by other labs in Drosophila, in the FMR1 knockout mouse, and our work as well in the FMR1 cow2 mouse show the rescue of behavior of Healx and rescue of different signaling pathways that have been altered in Healx. There are also evidence in the clinic in small studies where patients have been dosed with metformin and they have shown some improvement, and all this data have led to the two clinical trials that are currently ongoing on metformin, one in the US and one in Canada, with everyone is sort of expecting the result. That could be something quite encouraging. But what next? So one of the things for sure from this clinical trial, we will be learning a lot about the beneficial effects on metformin in the clinic or not, and also how translatable is the mouse model, and all the findings and all the preclinical evidence that we have so far with metformin, how translatable are there in human. And also we're going to be learning a lot around the different endpoints from the clinical trials. At Healx, we're currently expanding our preclinical testing because we already with metformin, we were able to validate our technology and benchmark the efficacy in the model. But we're currently exploring beyond that and testing additional repurposed candidates as single drugs and also in combinations. As they described before, we're using combination in a way that we can modulate several targets and several pathways at the same time. And together, if the drug synergize, we could achieve a greater effect in the mice and potentially in the clinic as well at some point. So I'm going to show you a couple of examples of combination that we've been working at the moment. So in this first combination, we are testing two drugs, Healx 001 and Healx 002. What we're doing here, we're testing these drugs at very low concentration when we normally see that these drugs individually are ineffective or partially effective in some of the behaviors in a way that we can sort of evaluate when we put them together if they are able to work in an additive or synergistic effect. So in here we test 4 different behaviors. For example, if we look at open field, we can see that the drug that actually is driving the effect is Healx 001, which is the one that actually is rescuing the open field test to the wild type levels. Healx 002 basically is not doing anything on its own because at its low concentration is ineffective. But when we put them together at low concentration, we still retain the effect that we observe with Healx 001, which means that if we put these two drugs together, at least there is no interference between them and we still retain the effect observed in the open field. If we look at the field conditioning test, the effect is the opposite. We can see that this drug number 2 is the one that is driving the effect here, which is able to rescue the field conditioning test to the wild type levels. Drug number 1, on the other hand, is not doing anything. It's not efficacious at this low dose. And when we put them in combination, we still retain the effect that we achieve with drug number 2. The interesting thing is when we look at sociability. The two drugs individually, Healx 001 and Healx 002, are ineffective completely in the sociability test. But when we put them together, we observe a rescue of this behavior to the same level of wild type, which is something really interesting. So potentially this drug could be working in synergy to rescue this behavior. And when we look at nesting, the effect that we observe is similar to open field. The drug that is contributing here is Healx 001, Healx 002 is not effective, and the combination is as effective as Healx 001 on its own. The other interesting thing is because we're using very low doses, we know that in this combination, we are testing Healx 001 10 times at a concentration that is 10 times lower than the current dose of this drug in humans in the primary indication. And for drug number 2, we're at least 2 times lower than the equivalent dose in humans. So we are going with repurposed drug and also with concentrations that are fairly low for how these drugs are currently used in humans. So it's something that is really encouraging as well. So here is another combination. Again, we are pivoting around Healx 001. In this case, I'm combining it with 001 and 003. And we can see that open field, same as we saw before, Healx 001 is the driver, 003 is not doing anything in this test, and the combination is still active. For nesting, it's the same, 001 is the active one, 003 is not doing anything, and the combination is still active. But in this particular combination, we can see that for field conditioning and sociability, actually what is working is the combination of the two drugs, because the two drugs individually 001 for field conditioning and 003 for field conditioning are ineffective. But if we combine them at these low doses, we rescue completely field conditioning. And similarly, we observe the same for sociability. So we are looking at a different combination where we can contribute differently with the two different drugs to rescue the different behavioral tests. And the last combination is just to show that not always things go as we planned. And you can see here that, for example, in sociability, the two drugs individually are ineffective. And when we put them together, in this case, we're not able to rescue sociability. So, which means that the way that they are working individually is not sufficient and these low doses to rescue this behavior. So in here, I'm showing you three different potential combinations that we've been working on that can rescue different behavioral tests in FRAXA to different levels and showing synergy of the drugs to some of these behaviors. And that leads me to my last slide. So it's around the conclusion and next steps. So I think I've shown today that Healx technology and the scientific expertise have identified several safe repurposed drugs that as potential treatment for Fragile X syndrome. The beneficial effect of metformin in this, the Fragile X cow-2 mouse model agrees with the preclinical data that has been performed by other labs, which is really, really encouraging. And we are conducting further preclinical validation of monotherapies and combination at the moment that is showing promising results, and we're characterizing that in behavioral tests and also trying to understand better how the combinations are working and try to get new insights of how we can combine repurposed drug better and find them efficacious in the mouse model. And all this preclinical data will be used to support Healx clinical development for Fragile X in the future. With that, I would like to thank our collaborator, the FRAXA Research Foundation, and also the lab GeneDDI that had done all the in vivo work in collaboration with us and the Healx team. So this is an old picture of the team. Now the team has grown quite a bit, but I don't have an updated image of the team at the moment. And thank you all for listening.
Thank you, Ivan. That was really fascinating. Really exciting for the future.
Thank you.
And just to remind people, save up those questions. You can start to register them in the chat function of Zoom, and we'll take them after our third presentation. And so with that, I'd like to introduce Dr. Anthony Hall, who's the Chief Medical Officer at Healx. And Tony will be presenting on the challenges in designing a clinical trial program for Fragile X. Tony?
Thank you very much, Mike, and thanks for the invitation. I hope you can see my screen okay. I'm not going to go into detail about our clinical trial design during this talk, mainly because we're still finalizing that at the moment, but hopefully you'll be able to see that in some detail on clinicaltrials.gov within a fairly short space of time. What I'm going to do instead is just to talk about some of the challenges that we faced in translating that fantastic preclinical data into the clinical development program that makes sense. So I guess there are sort of three main challenges that we've faced, which I've outlined here. The challenges of translating combination therapies into clinical trials. So that in itself is already quite tricky because with combinations you need to do some dose finding as you do with monotherapies, but you also need to demonstrate the relative contributions of the individual components within the combinations as well, and that's a regulatory requirement to do that. To some extent you can do that preclinically, but the regulators generally would expect you to be able to do that in the clinic as well. So, and I'll show you a little bit later that can get quite complex when you don't know your doses with your combinations. The other, then secondly, more sort of specifically, some of the challenges of running clinical trials in patients with Fragile X. It's not an easy condition to study compared to some conditions. And so we have to think about how translatable the preclinical domains are to the clinical endpoints. And so you saw from Ivan's presentation that there are a number of domains where the drug seems to be effective, but we don't know exactly how those translate into exactly which parameters in the clinic And that can then make it quite difficult to decide what the clinical trial should look like in order to try to reflect what we've seen preclinically. And on top of that, as you saw, some of the preclinical work, you see that it's quite sort of all or nothing with some of those parameters as well. So you don't really know whether you should, which of those doses you should go for to be able to achieve which effect. And then if you achieve an effect on one, will you also be able to achieve an effect on another parameter? So that's quite a challenge too. And along that theme, there's really a lack of definitive clinical endpoints for use in trials. There've been a lot of failed clinical trials in Fragile X, and so there isn't really anything that's perfectly accepted as a clinical endpoint for clinical trials in Fragile X. And then the lack of appropriate biomarker endpoints. I mean, it would be really great if there were some biomarkers that we could use that already had regulatory acceptability. But the lack of those makes it more complicated as well. And then finally, I think it's also worth mentioning the challenges of trying to mitigate for the COVID-19 scenarios. We're obviously looking at setting up a clinical trial in quite an unusual time, and we need to also think about how clinical trials will look in this new normal environment, and thinking ahead about whether there's anything specific that we need to do to accommodate that. So, what's so special about combinations? So I put this little factorial chart here. Just this is actually one that I've adapted from a design of a study with an antihypertensive, which of course this makes perfect sense for, because you can see with drug A and drug B, as you increase the dose of one and increase the doses in combinations, that you see an increasing effect. And this works really nicely with something like hypertension, where you can measure it in millimeters of mercury. It's a bit more complicated with a condition like Fragile X, where, especially if you're not even sure what the best primary endpoint is, how you're going to be able to tease out small differences in effect between doses, and whether, for example, two drugs used in low dose in combination might produce a similar effect to one of the drugs used at a higher dose, and how you're going to tease that out in a clinical trial is difficult. The other thing, of course, that is really important here is that there's a lot of groups. And so if you wanted to test 3 doses in a placebo, then you're looking at 16 groups. And within a rare disease, that really is completely unwieldy and not possible to design such a trial. And yet, we need to try to generate somehow this kind of information. So we were thinking, well, how could we do that? And one way to do it, which is not perfect, but it's a way to address it, is to look at fixing one of the doses. So drug A here, we fixed it at the high dose, and then we've looked at drug B, either placebo, so 0 drug B, low dose, mid dose, and high dose. And that helps, but you're still faced with a number of gaps in the information that you can generate from such a study. So, and 4 groups is quite manageable. So we thought, well, perhaps we could manage 1 or 2 additional groups as well. One of the things that comes out a lot in Fragile X studies is that there is often quite a high placebo response. And so we decided that we would add in a placebo arm as well to make 5 groups. On top of that, we thought for drug B, we don't have any information from this trial about what drug B does on its own. And that could be a problem, because depending exactly what kind of effect, if we see the effect as it's presented here, then it would be okay, it would be quite nice. But it could be that, for example, that that we selected the wrong dose of drug A to fix, and basically we might just see, everything might just come out the same. So we decided that we would add 1 dose of drug B as well. So we end up with a 6-arm clinical trial instead of 16 arms, which is much more manageable clinical trial, perfectly reasonable to do. It still leads to a fairly small number of patients per group compared to what we would really like, but it's a manageable clinical trial at least, and generates quite a lot of information from a reasonable number of groups. So then thinking about what specifically makes designing a clinical program for Fragile X difficult, you have to remember that the FDA requires substantial evidence of effectiveness, which normally means that you need 2 adequate and well-controlled clinical trials. They are a bit flexible, and they do say that this can sometimes be met by a single really good quality clinical trial plus some confirmatory evidence. So you might get away, for example, with a phase 2b and a phase 3 trial, provided you've discussed this and agreed it with the FDA in advance. But generally speaking, the point is that you need to provide substantial evidence of effectiveness. So that's what you need to think about at the beginning of the program. How are we going to do this? And even bearing in mind that it's a rare disease, you expect some flexibility from the regulators. You still need to generate the right amount of evidence and of proof. So just to, we obviously started thinking hard about whether there were biomarkers that we could use. And just to kind of refresh people's minds a bit on some of the terminology. So a biomarker is a characteristic that is objectively measured and is evaluated as an indicator of a biological process. And that's irrespective of any therapeutic intervention. A prognostic biomarker then is the biomarker that forecasts the likely cause of a disease regardless of treatment. And then a predictive biomarker is one that forecasts the likely response to a specific treatment. So, and then there are the clinical endpoints. So a clinical endpoint specifically is measuring how a patient feels, function, or survives. And a surrogate endpoint is a measurement that gives an early and accurate prediction of both the clinical clinical endpoint and the effects of treatment at that endpoint. And then the all-important thing is the validation, or the regulators normally call this qualification. So it's fine to come up with a biomarker, and it may even be quite a good one from a theoretical or scientific point of view, but it needs to be validated and accepted or qualified by the regulators. And if it isn't, then it doesn't really matter how good your data is on that biomarker. It may be quite difficult to persuade the regulators that this is a valid biomarker for you to use in order to show that your intervention, your treatment has been effective in that patient population. So we were thinking, of course, initially about possible clinical endpoints. And one of the things that we noticed immediately is that there are 67 interventional clinical trials listed in clinicaltrials.gov, and yet there are no approved therapies for Fragile X. So that tells you something about the success of those clinical trials and the endpoints that they've used. And so we obviously don't want to follow in that path, and we want to make sure that our clinical trial is successful. When you look at the clinical endpoints that have been used, and I've listed just a few of them there's a very large number of endpoints that are regularly used in clinical trials and in the clinic. And then a number of quality of life measures as well, the PEDS-QL, the EQ5D, the CARA-QOL. And these are also fine, but unfortunately they're not specific for Fragile X. And it is always useful to have, if you want to include quality of life instruments in your clinical trials, it is actually useful to have a disease-specific quality of life measure, because that makes it much more sensitive to changes that your treatment might induce in that particular patient population. So we noticed that recently NeuRen had published a study which had taken a bit of a different approach, and I'll go into that in a little bit more detail in just a second, where they'd used different efficacy domains looking at clinically relevant phenotype dimensions that are associated with impairment in Fragile X. And so we thought that seems like quite a good approach, because it allows you not to focus on one specific endpoint and hope that you've chosen the right one. But the other thing that is really important to consider, of course, in any clinical trial, is how to keep the number of assessments to an acceptable level. So it's fine to have, you could just say, okay, let's measure everything. But unfortunately, if you do that, it means that it's really, really uncomfortable for the patients and for the carers. It all takes a very, very long time. And then you run into the issue that you're probably going to end up missing some data if you try to impose such an intensive assessment on patients. So we need to find an endpoint that is most likely to be influenced by treatment. And we also need to take into account potential confounding of other treatments as well. So I'm not going to talk about this or the next one in any detail. And I show them just really to show that this was a publication from 2013, which looked at some of the endpoints that have been used in clinical trials at that time. There's probably even more now, 7 years later. But just to show that there's a lot of different endpoints that have been used. And you can organize them to some extent into these domains, behavioral, cognitive, and the biomarker domains. And that's exactly what the approach that Niren did. So what Niren had done is they'd organized this into 4 clinical domains and a biomarker domain. And then they developed their own separate Fragile X rating scale and their own Fragile X visual analog scale as well. So within each of these domains, they've organized these into core and secondary assessments. And then the core ones are the ones that are scored for the primary outcome. So this is quite a clever way of doing it, because what it means is that you can, instead of having to pick one and focus on that and hope that you've got it right, you actually get a score for all of these. And they score it basically plus, 0, and minus, depending on whether it's improved, stayed the same, or got worse during the course of the clinical trial. And then the statistics is able to look at that and determine whether there's an overall effect. And so this is the approach that we're adapting for our clinical trial. Unfortunately, that Fragile X rating scale that was developed by Niren hasn't been published yet, so we can't use the exact scale, but we will use something along those lines, together with some of these other endpoints that are mentioned here and that have been used in other clinical trials. So there are some good pros to doing this. Like I said, it safeguards against missing that crucial domain by selecting the wrong endpoint. It may be that your drug works, but if you've chosen the wrong endpoint for your clinical trial, then you won't see it. And that may well be what's happened with some of the other, some of the failed clinical trials along the way. And it's a pretty good approach for a proof of concept or pilot trial. Unfortunately, it's not very good for a registration trial. And the main reason for that is that the study almost certainly will be underpowered in terms of reaching statistical significance. If you think about what I mentioned about having to do some dose finding, having 6 groups, 6 treatment groups in your clinical trial, together with an endpoint which is essentially a composite, the chances of you getting a statistically significant result from that are pretty low. And so, but on the other hand, it does generate really, really useful information on which of those domains are most likely to be the ones where the drug is working. And then you can take that and focus on those for the next clinical trial. The other thing is that many of these assessments themselves are already composites. And so, as I mentioned before, we have to think really carefully about the amount of time that it takes to do the assessments and make sure that we can capture as much data as possible, but still keep it comfortable and reasonable for patients and for carers. And we also have to make sure that we don't run the risk of having too many missing values at the end. We also looked at potential biomarkers, and one of the ones that interested us from the beginning was the EEG, and in particular the gamma power on the EEG. This is actually from a mouse study, but it shows in the graphs on the right-hand side there, you can see that the power is much higher. That's the blue curve is much higher in the knockout mouse than it is in the wild type mouse. And so we thought maybe this would be an interesting one for us to use in our clinical trial. This actually is from the same paper and presents more or less the same information, but presents it as a ratio. And again, over there on the right, you can see that there seems to be quite considerable increase in the gamma power in the knockout mouse. So we thought, great, that looks like a good one for us to choose. But then we started looking for some human data, and there's not much in the public domain. In fact, I think this is the only publication that we could find. And here you see that there is an increase in the gamma power in the Fragile X patients, but the increase is maybe not as much as we'd seen in the mouse data. And we then became aware that there is some more data as well, but unfortunately it's not been published yet, but we did get a preview of this. And when we looked at that in detail, we saw that the increase in gamma power was, yes, there is an increase in Fragile X patients, but there's quite a spread as well. And we felt that if we wanted to try to concentrate on patients who had sufficiently increased gamma power, that it would mean that it would make it very difficult for us to find and select patients for the clinical trial and make the clinical trial pretty much unworkable. So in the end, we decided that this was not a reasonable endpoint for us to choose, coupled with the fact, of course, that this isn't a proven surrogate endpoint either. So we would be taking a big risk if we were to select this one and run the risk that we didn't show anything, and even if we did, that it's not accepted. So finally, I just wanted to touch upon some of the issues that we've grappled with around COVID-19 and how that could affect things. So many clinical trials have really ground to a halt during the lockdown. And I mean, we're quite lucky that we hadn't started ours really, because it could have been very difficult if we'd started the clinical trial and then everything had to be stopped due to the lockdown. So one of the things that we started looking at was whether we could mitigate some of these risks by using a kind of decentralized clinical trial approach. For example, there are a lot of companies now that are advocating using app-based assessments, wearable technologies, remote video consultations, for example, and even home visits for assessments so that the patients and the families don't have to travel to the hospital. But the problem with that is that the kinds of things that we want to measure in a Fragile X study are not easily amenable to this. This kind of technology would work really well with something like an acne study, for example, where you could simply give the patient the mobile phone with the app on it, and every month they would take a picture of their acne, upload it to the server, and it could be interpreted by an AI or by an independent expert. That would be really perfect for a decentralized approach. But with the kind of complicated assessments that we want to do in our Fragile X study, it's actually much more difficult to do that. So in the end, we've had to compromise a bit, and even though we've managed to make some things remote, we won't be able to make everything remote. So just in summary then, as I've said, I think developing treatments for Fragile X syndrome is pretty challenging due to the difficulty in identifying appropriate clinical endpoints and the fact that there's no reliable biomarker endpoints that have been characterized. But with the use of a composite endpoint with domains, we hope that we can overcome some of those difficulties. We also have to factor in the challenge of developing combination treatments, which adds an extra level of complexity. And then in the current situation, of course, we've also had to contend with how to mitigate against the impact of COVID-19. Thank you.
Thank you, Tony. That was really, really great. And if I could summarize the three presentations so far, Dave started out talking about our quest for greater simplicity, and Yvonne talked about using the mouse model to try to validate some of these combinations. And then you've described the effort to take this into clinical trials. And in the end, the result is the more we look for simplicity, the more complicated things get. So that's my takeaway from today. We're still searching. So I just want to remind everybody that you can ask questions in the chat functions. And I thought, since we have a large number of folks from Healx on the call as well, if you'd like to invite some of the other folks from Healx to participate in the questions and get a little bit more back and forth going, I think that would be very appropriate in this situation. But I'll just start by throwing out some of the questions to our speakers. And specifically referring to Dave's presentation, did you publish your discovery before patenting? Is it normal? Is this a normal process?
The neuropharmacology paper was published after we'd filed patents.
Okay, and another question, what do you think about the new medication, lumateperone? I have not used that one. Anybody know anything about that?
Eric, do you want to unmute people?
I'm now able to unmute myself. This is Bruce Bloom from Healx. So this is a new drug that's just come out on the market for schizophrenia and perhaps for some other mental health diseases. I don't know that we've done any work on it yet, but it would be an interesting drug to think about for Fragile X, but I don't think we're looking at that quite yet.
Okay. Dean Clark asks, have you considered a double-blind model to measure or validate knockout mouse activity?
Yeah, well, actually the experiments in the mouse are currently, the CRO is doing them blinded to sort of make sure that the experiments are robust enough.
And that's generally true for most of the in vivo work in the mice, that the people who are evaluating any of these behaviors are doing it blinded, and they don't know the genotypes of the mice or their exposure history to the drugs, that the drugs are given by somebody else. And so it's all blinded by the people that are evaluating it. Okay, how reliable and predictable, oops, excuse me. Oh, how many trials are underway on metformin? Are they still recruiting? How are they going? What's the dosing? How does the dose compare to diabetes dosing? I can just answer some of that fairly quickly, in that none of the metformin studies, the controlled trials, have been completed yet. They're all still ongoing. You can look at clinicaltrials.gov to see where the different clinical trial sites are. Generally speaking, they're using diabetes type doses of metformin in these trials. Anybody else have anything they want to add on metformin trials? No? Okay. Osman asks, how do you select the drug, and do you test the transcriptome after drug use? You plan trials in mainland Europe, and could variability in drug efficiency in different patients, how to test? Yeah, so do you use transcriptomics as an outcome measure? I guess that's the first question.
Yeah, I can take that one. So this is something that we are currently doing right now. So after we just did sort of the screen in the mouse to come up with different combinations that are interesting, now we're trying to understand how the combinations are working and if they're actually reverting the transcription of genes towards wild type levels. And we're currently performing those RNA-seq experiments to understand how the drugs are working better.
And of course, the issue in humans is that you always have this discrepancy between blood and brain, and ideally what you'd want to see, at least we think what you'd want to see, is a normalization of transcriptomics in the brain. You obviously can't do that in human clinical trials, outcome measure, but you can do that in mice.
Yeah. So we are sort of collecting different regions of the brain and performing RNA-seq from the different regions to understand this a little bit better.
I addressed the one about mainland. I think the same person has asked about trials in mainland Europe. So the answer to that is yes, but not for this study. So this first study is quite a small study and will just run in the US. And unless the US gets locked down again or, you know, whatever, something unforeseen, but the plan is to run the study in the US. But for sure, for subsequent studies, we'll definitely bring those to mainland Europe.
How long do the beneficial effects of the combination drugs last? And I think we're talking about a sort of carryover effects. In other words, can you take these drugs and do they keep working after you stop taking the drug?
Yeah, that is a really good question. That is something that we haven't done yet. So washout effect. So we normally have conduct all the experiment dosing for 2 or 4 weeks and then performing the behaviors right after. But it's a really good question. We need to address that at some point, to stop the dosing and wait and then do the behaviors to see for how long we can still retain the effect. Really good question.
I would add that we have done some studies of carryover effects in the animal models, and we have indeed seen pretty significant carryover effects with some drugs in the mouse model. We also routinely see that in psychiatry, and maybe one of the ways of predicting that is if you really can see rescue of transcriptomics, then you know you're having a long-term effect in the brain. And that's what you see with a lot of the medications that are on the market for psychiatric indications. Heidi wants to know, do the drugs being tested have anything in common, such as blood sugar management?
I'll have a go at that. I mean, metformin is used for diabetes and it does manage blood sugar, but it's probably the other mechanisms of the drug. It's a drug, I think, with 20, 30, 40 different effects, different mechanisms. I mean, it's being trialed in Boston as an anti-aging drug at the moment. It's a very complex pot of mechanisms of action. The second drugs we're adding, you know, when we put combinations of drugs together, we're generally finding what works and then working back to the mechanisms, and the mechanisms really don't necessarily relate to the original mechanisms for which the drugs were developed. So yeah, generally they have nothing in common really. Ivan, do you want to add to that?
Yes, so for the 3 different combinations that I've shown, they're completely unrelated of blood pressure drugs, and they have completely different mechanism of action on target, but also it could be some secondary pharmacology there that we don't know yet. So that's one of the things that we like to find out with the RNA-seq experiment, is to try to understand if it's just on target effect or potential other secondary pharmacology that could be driving the effect as well. But they're completely unrelated drugs.
I think it's worth adding that even for drugs that are 20 or 30 years old, we still don't fully understand all the mechanisms by which they work. There are new scientific publications every year on drugs identifying new mechanisms.
If I could just add a follow-up question to that. One of the recurring themes that we see in repurposing for Fragile X, whether it's by Healx or just our sort of rational repurposing efforts that we've made, is that a lot of the secondary mechanisms of action with a lot of different drugs that have been identified is anti-inflammatory. Would anybody like to comment on that? Is there a role for anti-inflammatory treatments in Fragile X in general?
Well, personally, Mike, I'm not aware of a mechanistic link.
Yeah. No, it's funny that a lot of things seem to have a primary mechanism in one direction and a secondary mechanism that's anti-inflammatory. Minocycline, lovastatin, a number of others, even metformin. These have been suggested, but how prominent they are in actual use, I don't know.
Yeah.
Okay. How reliable and predictable are the behavioral tests in knockout mice compared to patients? question.
Well, that's the big question, of course, that we
Yeah.
need to address with the clinical trial. And so yeah, I think I mentioned this as well during my talk that that we don't know at the moment, and that's also one of the reasons why it's quite risky to be too definitive about which parameter you try to. If there's one parameter in the model that looks particularly good, if you choose that one as your primary endpoint, it might not be the right one to choose because it might be different in humans. And the same thing with doses as well. The model is quite sensitive, and so you could easily choose the wrong doses. And so that's one of the reasons why we need to do some dose finding in the humans too. So so that's what we aim to find out in the clinical trial, whether the drugs work in humans.
Maybe I can add something that Neuren's compound, for example, that is one of the ones that is going ahead on clinical trials, doing quite well, was tested in the mice, and the same other compounds are already in clinical trials, like Ovid's pharmaceutical as well. They were all tested in the mice. So obviously it's not the same that in humans, and the translation, you know, obviously will be relative, but so far it's not that, I think it's relatively predictable using mice in some aspects, not all, of course.
And I just wanted to mention Steve Warren's famous quote is, "There's never been a better time to be a Fragile X mouse," because just about everything seems to work in the Fragile X mice. Although, as we've seen, that's not really true. If you actually refine your doses, you can find dose response curves, and you can find subtherapeutic doses of even the most effective treatments.
Yes, we found loads and loads of compounds that didn't work on Fragile X. Unfortunately, negative data is not published, so you know, that remains in our knowledge, and people do not know how many compounds have failed. But some are gone through, like the Neuren's compound, and it's doing quite well in clinical trials, and others didn't, but many have failed too.
Okay, next question. Tanya, did you publish your discoveries before you patented them? I think they answered that. So what was your approach to identify new drug candidates from Ivan's presentation?
Yeah, so the majority of the candidates that we've been testing for Fragile X come from the method that rely on transcriptomic of the disease and the drugs. But we currently have 3 other methods that are independent of transcriptomic that we also could apply for Fragile X as well. But today, the method that has been more successful in Fragile X is the method that is relying on transcriptomic.
And also for you, Ivan, could you talk about the effect of metformin on dendritic morphology? Was this done in adult mice?
Yes. Yeah, that was done at the end of the experiment. So there were mice that were 2 months old, and they were dosed at 100 mg per kilo twice a day with the drug. And yeah, so that was how the
4-week IP, or was that from the continuous infusion?
4 weeks IP. 100 mg per kilo twice a day.
Twice a day, yeah. Richard Novak asked, do you plan on using patient transcriptome signatures as a predictive biomarker in trials? If so, what does the FDA think about this?
No, what we're planning to do is, what we're doing now is on the mice to understand better how the combinations are working, but I don't think there's any plan to use this in the trial, Tony.
No, we don't have any plans to do that.
Very expensive, and it would only be in blood.
Yeah.
And also, you could only prove it's predictive after you've got efficacy with a drug and you've done the transcriptome in parallel, so it wouldn't work. It wouldn't be predictive for us in this initial trial.
Osman asks, can't you use improvement in cognitive ability over 3 to 6 months as an outcome measure?
Yes. So So that will be part of that composite endpoint. The issue is that we don't want to focus on one thing, because we don't know for sure which particular endpoints we're most likely to be effective. So we want to try to have a composite where we can look at several different outcome parameters and try to figure out from this study which is the most appropriate one for us to use then in the subsequent study.
And I would just add that there's a lot of excitement about the NIH Toolbox, which is a kind of a rapid cognitive assessment that's specifically designed for developmental disabilities. Conventional IQ testing, as I think we all know, is pretty much worthless in Fragile X, and that's been used in previous clinical trials with no success at all. Gordon Wong wants to know, how did you determine which drugs to use for the combinatorial trials? And I guess optimization of the combinations is the real question, right? Not just coming up with any number of drugs that you could try in combination.
Yeah, it's very complicated. The combinations that we're taking to clinical trials, of course, were validated in the mice. And how do we select them for the mice? We started with single drugs. We ran algorithms that included those single drugs in combinations to see how they might rank. And also, we looked at the mechanisms of the drugs and tried to figure which might actually stand a chance of having additive or synergistic effects. At the moment, it's quite difficult to do that. And I think the success rate of combinations that were synergistic, I don't know what it was, but it was quite low. It's probably only 20% or less. Ivan, I don't know whether you've ever counted. It's pretty tough to come up with synergistic combinations.
Yeah, not that many become completely synergistic and rescue. When we start doing a scoring of the combinations to come up with the better ones to progress, and some of the combinations fail because of you lower the dose to a point where they become suboptimal and they don't rescue the behaviors anymore, which is something I think is also good to have, to show that it's not always working and you can have sort of negative results as well to compare with.
Yeah, and we've seen the opposite effect at times where two drugs that work individually, when you put them together, they stop working, they antagonize each other. So you get sort of neutral effects, positive effects, and negative effects with combinations.
Anyone want to comment on efforts to repurpose for Down syndrome, trisomy 21?
It's not something we're working on, no.
Bruce?
Why the link?
When I was at Cures Within Reach, I know there were a couple of things that we looked at, none of which we thought had the kind of preclinical support that made them worth taking towards clinical trial. So I don't know of anything right at the moment because we're not looking at it as Healx, and I haven't checked back with Cures Within Reach recently, but maybe Osman could contact Cures Within Reach to see if they're working on anything for trisomy 21.
Okay. And if everything goes according to plan, when will clinical trials start?
Okay.
And where will they start? I think.
And where? So yeah, I think I already answered the second part of it by saying that the first clinical trial will be in the US. And if everything goes fine, then we should be able to start in the first quarter of next year. So we aim to get our IND in this year, and then first patient in the first quarter of next year, if all goes smoothly. You never know with regulators.
And Matthias wants to know, what are your thoughts about a combination therapy for Fragile X consisting of something like fluvoxamine, aripiprazole, minocycline, and N-acetylcysteine? That sounds crazy. Who came up with that? Oh, wait a minute, that was me. Any thoughts on that?
No, I mean,
I would just urge caution because, as I said, much of the time when we put two drugs together, we don't get synergy or even additive effects. You can get antagonistic effects. It's very, medicine really doesn't understand combinations particularly well at the moment, and yet unfortunately physicians are having to give multiple drugs to control, for instance, high blood pressure or diabetes. And once we start mixing classes of drugs, the research just hasn't been done. So I think we have to be very cautious about giving 3, 4, 5 drugs to patients.
I think in case anybody doesn't understand the inside joke here, I recently wrote an article for the FRAXA website that was about my favorite combination, which was fluvoxamine, aripiprazole, minocycline, and N-acetylcysteine. And I talked about all the studies that have actually been done of various combinations of those drugs. So those are all drugs that have been used together in clinical trials and clinically fairly extensively. And that particular combination appears to work quite well in Fragile X, although not necessarily advocating that everybody immediately run right out and start all 4 drugs all at once, that's for sure. Gordon Wong asks, can CSF measures be used in clinical trials? So can you tap into that spinal fluid?
Yes. There are clinical trials that do that, but obviously there needs to be some reason to do it, and I'm not aware of any biomarker endpoint that we would be that would be useful for. So I can't really see a role for it here in a Fragile X study, but of course in the right clinical trial that can be done. It's not very comfortable for patients, obviously, but if it's needed, then it can be done.
Talk about hurting recruiting. That would really.
Yeah.
Charles.
Hi, Mike.
Oh, I'm sorry. Go ahead. Is there somebody else?
Hi, Mike. It's Julie at Healx. I was just going to comment on the CSF piece. We've been exploring. We're not going to use it in this trial, but the use of sort of CNS-derived exosomes could be really useful in clinical trials in the future in Fragile X to measure biomarkers. So, that circulating exosomes, which are derived from the brain, you can actually extract from the peripheral blood. So that's a great potential technology for use in Fragile X translational studies.
So it's sort of like getting a brain sample, but just by taking blood.
Yeah.
Yeah, area of great interest in autism studies in general, I think.
Yeah.
Charles Chamberlain wants to know, is there any anecdotal information about the effects of metformin in Fragile X cases where the drug's being used for diabetes treatment? So just incidentally being exposed to metformin.
I don't know the answer to, I'm guessing that probably, Mike, you know the answer to that better than me. I'm guessing that probably the first idea came from an anecdotal use, but somehow it's got into 4 clinical trials now. So, and obviously that's the proper test of it. But I'm guessing that the idea must have come from a chance finding anecdotal information originally.
Well, oddly enough, it didn't. And the interesting thing is FRAXA has now come up with a number of repurposed drugs through this kind of rational drug discovery method. And each time when we come up with an available drug that's a potential candidate for treatment in Fragile X, a number of clinicians will come out of the woodwork and say, I had some patients who took this and they seemed to do really well, but none of them ever did it ahead of time. So that kind of retrospective observation is always a little bit suspect. But nobody ever came up with that observation in Fragile X. And Liz Berry-Kravis, who's one of has one of the largest Fragile X clinics, has made the observation that even though a lot of Fragile X patients get treated with atypical antipsychotics, which are notorious for causing metabolic syndrome and type 2 diabetes in people who take them, especially kids who take them, she doesn't see Fragile X patients developing metabolic syndrome and type 2 diabetes. So paradoxically, there seems to be less of a need for metformin treatment in Fragile X. And also a similar observation that even though lovastatin has been implicated as potentially therapeutic in Fragile X patients actually have lower than average cholesterol levels. So the likelihood of being exposed to lovastatin just in the normal course of things is actually lower for Fragile X people. So in neither of those cases were any independent observations ever made. Okay, I got another question for Ivan. Have you tried the chronic treatment on juvenile mice? If yes, did you manage to prevent different behavior phenotypes appearing in adulthood?
Yeah, no, we have done it always in adult mice around 2 months old. My understanding is that the characterization of the battery of behavior was done in adult mice, and where the majority of the behavior show differences between the knockout and the wild type. But it's a really good question. So I have seen some publication where people also use younger mice for some other kind of test, but we haven't done that.
And one of the practical difficulties of using any kind of chronic treatment in younger mice is that the mice don't get weaned until a certain point. So in fact, when Irina et al. Wanted to test minocycline in young mice, she had to actually give it to the mothers and give them the dose via the milk. Osman asks if metformin is used as an anti-diabetes drug, so why is it difficult to perform clinical trials on Fragile X patients?
Yeah, I think that really just comes down to the complexity of doing studies in Fragile X specifically. And so it's not a property of the metformin, it's a property of the Fragile X that makes it difficult to do studies in terms of what are the most appropriate endpoints to choose, and what's the best way to demonstrate that your treatment works if it really works. And that's more difficult in Fragile X than it might be in some other conditions. But clinical trials are always complicated in terms of choosing the right endpoints.
And I guess a follow-up question from Osman, and let me just paraphrase this. If we look at transcriptomics in the mouse and we find that you see really good effects in brain and they correlate with blood effects in the mice, does that make it look better for use in humans?
Yes, I guess it could be a surrogate, analyzing and comparing the blood from the mice and the brain and see if they sort of look the same, and then we can sort of move that into potential use in humans. So it's a good suggestion. It's something that we could consider.
Yeah, or the exosomes. I guess you're not going to get transcriptomics from exosomes. But... Well, if there are no more questions, I'd like to once again thank our speakers. Really fascinating discussion. And we really appreciate your sharing with us the challenge of trying to do this kind of innovative study in Fragile X. It's not easy. And I joke a little bit about how the search for simplicity has led to more complexity, but it really is true that we try to use available drugs and we try to take what we've already got and make it work better. Sounds good, but it's not easy. So thank you all for working on this, and thanks for sharing this struggle with us.
Thank you.
Thank you for
having us. It's been a real pleasure to work with you and FRAXA, Mike.
Yeah, thanks, Mike. And thanks to everybody who was listening for all those great questions.
Yeah.
All righty. Well, thanks very much. This concludes our presentation. Bye-bye.
Bye-bye.
Bye.